what's going on, guys? Tonight we're going to be talking about a very important topic. We're going to be talking about the proximal convoluted tubule. Now, let's keep in mind that this is all part of renal physiology. So, as we all know, whenever the blood is filtered by glomerulus, the first part of the tubules that it gets down to is the proximal convoluted tubule. Now, you guys might be wondering why I already have this drawn. This first right here, the amino acids and sodium amino acids, and the sodium and glucose. This is a representation to tell us that 100% of amino acids and 100% of glucose is reabsorbed back into the proximal convoluted tubule cells, which is then reabsorbed back into the blood. And as we can see right here, this is the lumen, this is the urine, this is the urinary side basically. So it's coming down here. So, and then it gets reabsorbed by the proximal convoluted tubule, right? And then after that, it gets reabsorbed back into the interstitium, which gets back into the blood. Um, we need to keep in mind that about two-thirds of water, two-thirds of water that gets into this proximal convoluted tubule is reabsorbed. And this happens by an osmotic gradient basically. So sodium chloride is also reabsorbed basically. And whenever we have this sodium chloride reabsorbed, what follows this sodium chloride essentially is the two-third of water of H2O that gets into the proximal convoluted tubule. So far we've said 100% of amino acid is reabsorbed. We've also said that 100% of glucose is reabsorbed back into the proximal convoluted tubules. And now we're also saying that two-thirds of water is reabsorbed back as well. Okay. Now, as we can see in the first diagram, as far as like the sodium amino acid, this is a symporter. And what I mean by that is pretty simple. We can see, I'm just going to draw with a different color. We can see that this is going in the same direction. Okay, that's what I mean as a symporter. This is all going back into the proximal convoluted tubular cells. Now, that's a very important point. And as we can see as well over here, it's all going back into the same direction. Okay, now we might be wondering right now that, okay, how come we keep having sodium? I mean, it's only so much sodium that a proximal convoluted tubule should be able to take. But if we go back to this side where we have the blood, the interstitium side, we're going to see that we have this pump. This is a sodium potassium pump. And we're going to see that for every two potassium that we have coming back into the cell, we are able to pump out 3 sodium ion. Now, what that makes is this, what that causes is that it causes a very low intracellular sodium concentration. If we have a very low intracellular sodium concentration, the sodium here on the urinary side, the sodium here is able to then easily go into the proximal convoluted tubular cells. Basically, the sodium in this urinary side basically is able to go into this proximal convoluted tubular cell. Is able to go into this proximal convoluted cells because we have a low gradient of sodium, and we have this low gradient of sodium because the sodium is being pumped out by this ATP pump. Okay, so that's a very, very important point as well um, for us to also know basically. Now, another thing that is being absorbed by the proximal convoluted tubular cell is phosphate. Now, phosphate is being reabsorbed by proximal convoluted tubular cells. We need to keep in mind that phosphate is regulated by PTH. Now, PTH is the parathyroid hormone, right? Now, we can also call PTH, so I like to call PTH a phosphate trashing hormone as well. Phosphate trashing hormone. What that means is this: when the PTH level is high, it blocks the reabsorption of phosphate. Okay, so phosphate is not reabsorbed basically. When the PTH concentration is high, phosphate is not reabsorbed. Okay, now this is very important because we know that phosphate is important for the regulation of calcium, all right, in the blood, calcium and phosphate. So whenever we have increased PTH, phosphate reabsorption is blocked. When phosphate reabsorption is blocked, now we can easily just pee out phosphate. Basically, phosphate goes down into the urine, and that way we can get rid of phosphate. Okay, so I'm just gonna draw these right here so we don't forget it. We can kind of, keep that in mind basically. So let's just kind of assume that this is the pump. So now another thing that we also need to keep in mind that is reabsorbed by these proximal convoluted tubular cells is chloride as well. We also need to keep in mind that uric acid, potassium is also reabsorbed, okay? And now a very, very important point is needed to be made. Now, as we can see so far, we've been using the same porters for this reabsorption, for the reabsorption of pretty much anything that we've talked about, right? Now, this is gonna change right here because we're gonna have hydrogen ion go out, but as we can see, this is more of an antiport. It's going in two different direction. Okay, sodium is going in, hydrogen is going out of the cell. Now, this is very, very important for hydrogen to be going out of the cell. There is a particular reason for this, for us to have this. Now, in order to create space, let me just kind of erase this so we can really understand exactly what I'm talking about, right? So we take this phosphate again, we're just gonna push it back into the cell. Okay, so we got that point. So the hydrogen that we're talking about again, let me redraw this. Sodium is going into the cell. Sodium is going into the cell. Hydrogen is coming out of the cell. Now this is hydrogen, right? Now we might ask, why is this needed? This is needed for the reabsorption of bicarbonate, okay? This is needed for the reabsorption of bicarbonate. In order for us to reabsorb bicarbonate, we do need hydrogen. Now, about 80% of bicarbonate in the urine is reabsorbed by the proximal convoluted tubular cells. And this happens by this reaction right here. Whenever we have hydrogen and bicarbonate here, they both attach together, okay? Now this is gonna just give us H2CO3. Now, as we all know, carbonic anhydrase is gonna be involved in this reaction. With the help of carbonic anhydrase, we're gonna be able to generate CO2 plus H2O. Now, these two can easily just diffuse through and get back into the proximal convoluted tubular cells, right? Now, once they diffuse through, we have CO2 plus H2O, right? Now, with the help of carbonic anhydrase again, and I'm spelling that as just Ca, we are going to be able to generate H2CO3 one more time. And then this H2CO3 can then easily split into hydrogen ion, right? And bicarbonate. Now, this makes perfect sense because we're just we're literally starting this whole equation with this hydrogen, and we use this hydrogen to be able to attach into these, and we're just able to, you know, with the help of carbonic anhydrase, we're able to generate CO2 and water, and the reaction just continues like that, basically. Now, luckily enough for us, we have a pump. We have a bicarbonate pump right here, basically. So this bicarbonate is then able to go back into the bloodstream, and this is very, very important for acid base. Basically, this is very, very important. This is used for the regulation when it comes to stuff like metabolic acidosis or metabolic alkalosis. And one thing that we need to keep in mind is that this antiport right here is actually stimulated by angiotensin 2. by angiotensin 2. Okay, now this is very important